Hi and hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Bioneet. In the last video we were discussing goals and methodology of Human Genome Project. Still the salient features and applications of Human Genome Project are in pending. So today in this part of videos of molecular basis of inheritance, we are going to talk about those salient features and applications of Human Genome Project. Before we going to start this, here a small request to kindly watch all my videos made on this molecular basis of inheritance for the better understanding of the concepts. And one more request is please like and share the videos and subscribe my channel. Let's get into our today's topic, starting with the salient features of Human Genome Project. The Human Genome Project has revealed some interesting facts and numbers regarding the human genome. Now we are going to discuss those interesting facts and numbers of human genome under the heading of its salient features. Earlier, the scientists of Human Genome Project expected that the human genome would have 80,000 to 1,40,000 genes, but actual number of genes in the human genome are much lower than these previous estimates. Finally, the Human Genome Project has revealed that the human genome consists only 30,000 genes. This is the fact revealed by Human Genome Project. And then this is the 3,164.7 billion base pairs that the human genome consists, which means the total number of base pairs in the human genome are 3,164.7 million base pairs, are simply 3.1 billion base pairs. Then this is 3,000 base pairs is the average length of the genes in human genome. You know very well that the length of DNA or the length of gene measured in terms of base pairs. 3,000 base pairs is the average length of genes in human genome, but the length of genes in human genome varies greatly. The largest gene identified by the Human Genome Project is the dystrophin gene in human beings. What is that? Dystrophin gene. The length of this dystrophin gene is measured about 2.4 million base pairs. This is a fact revealed by the Human Genome Project. Human Genome Project also revealed that 99.9% of the genetic makeup in the individuals of a population is similar. Nothing but 99.9% of the genetic makeup in all the individuals of human population is similar, but each and every human being differs with the other human being just by having 0.1% different genetic makeup, which means the each and every human being is distinct by its genetic makeup, though we are all belongs to the population of human beings. So we got a great information regarding the human beings. Among all the sequenced DNA of human genome, about 50% of human genome is functionless. Functionless means non-functional DNA. This non-functional DNA of about 50% is otherwise called junk DNA, which do not involve in the synthesis of any structure. The Human Genome Project has also revealed one more interesting fact, that less than 2% of the human genome is involved in the synthesis of proteins. How much genome involved in the synthesis of proteins? Only less than 2%. And the Human Genome Project also revealed that the repeated sequence makes up a larger portion of DNA in human genome. The repeated sequences are nothing but the satellite DNA. What is this satellite DNA? The DNA which is highly condensed. We have already discussed in the last video about the satellite DNA and its types. Highly condensed non-coding repetitive sequences. What are these? Repetitive sequences separated upon centrifugation is said to be as the satellite DNA or repeating sequences. These repetitive sequences repeats for about 100 to 1000 times in the human genome. These repetitive sequences very much useful in DNA fingerprinting. Why? Because the number of repeating units of each repeating sequence is unique for every individual in the human being. Some individuals of human being may have only 100 repetitive sequences of a particular repeat, and some individuals may have 1000 repetitive sequences of that particular repeat. Some individuals of human being population may have 500 repetitive sequences of that particular repeat. So the number of repeating units of each repeating sequence is unique for every individual. Based on this principle, we are using these repetitive sequences of satellite DNA in identification or tracing of culprits in crime situations. To that we are going to discuss in the next video in the lesson DNA fingerprinting. And these non-coding repetitive sequences, though they do not involve in the expression of RNA and proteins, but they influences the chromosome structure, dynamics and evolution. This is what about the salient DNA or repetitive sequences we came to know through this human genome project. And then the chromosome 1 is the largest chromosome, having about 2968 genes, and it also said the smallest chromosome in the human genome, that is Y chromosome. The Y chromosome is having only about 231 genes. Here we told that human genome project sequenced only haploid set of chromosomes. Haploid set of chromosomes means how many chromosomes comes in haploid set of human beings? 23 chromosomes. But the 23rd pair of chromosomes in human genome are allosomes, which is a heterologous pair where X chromosome is different, Y chromosome is different. So that human genome project has sequenced the 23rd pair completely. Therefore, the number of chromosomes sequenced by human genome project are 22 plus X and Y chromosomes, total 24 chromosomes. Keep remember this. The last fact we got from human genome project is there are 1.4 million SNPs. Here SNPs means single nucleotide polymorphs. What is SNP? Single nucleotide polymorphs of DNA. What is mean by the single nucleotide polymorphism? Several nucleotide sequences, they looks almost similar in the sequence of nucleotides, but they differs by one nucleotide. Say suppose for example ATGC-TCA is a sequence, AUGC-TCA is another sequence, ACGC-TCA is another sequence. These are the different sequences of DNA which almost looks similar, but they differs by a single nucleotide. Here at second position it is having thymidine, it is having uridine, and it is having cytidine. These DNA sequences differing by a single nucleotides are called single nucleotide polymorphism. This is a polymorphism of DNA where the DNA sequences are differing by a single nucleotide. Those are called SNPs. These SNPs in the human genome are about 1.4 millions. This entire information, whatever we got from Human Genome Project, promises us to revolutionize processes in finding out the chromosomal locations for disease associated genes and in tracing of human history. Now let us move on to the last concept of a Human Genome Project, that is applications of a Human Genome Project. This 13-year project is actually a study of human beings or study of biological system at a molecular level. Means it provided the clues to understanding of a human biology. Therefore, the results of Human Genome Project has influenced the human life in these modern days, especially in the field of medicine. This Human Genome Project has provided the knowledge about a diagnosis of disease by identifying the defective genes in the diseased individuals. How can we diagnose the disease by using this Human Genome Project? That is very simple. Just by comparing the defective genes of diseased individual with the normal gene of healthy individual, we can come to know whether the individual is diseased or not. Having the knowledge of sequencing of genomes, till now over 30 defective genes have been traced out in the human genome associated to the diseases such as breast cancer, muscle diseases, deafness and blindness. So this is what we have achieved through this Human Genome Project. Along with the diagnosis of disease, the Human Genome Project also helpful in understanding the base for new technologies in treating of disease. Such a new technology developed in the field of medicine through this Human Genome Project is gene therapy. Gene therapy is the new method of treating of diseases. What is this gene therapy? In gene therapy, generally we can replace the mismatched base pair in the defective gene of a diseased person with a correct base pair, so that repairing of a particular gene responsible for a particular disease results into a curing of disease. We have been curing the diseases through this gene therapy, which is the outcome of Human Genome Project, such as cancer and some metabolic disorders. Therefore, the Human Genome Project, besides providing the knowledge about diagnosis of disease, it also provided the knowledge in developing of new technologies such as gene therapy in curing the diseases. And the next application is the completion of non-human genome project. This Human Genome Project has led to the sequencing of genomes of non-human organisms ranging from prokaryotes to the plants and human beings. The non-human genome project is still be completed are genome project of bacteria, which is a prokaryote, the genome project of yeast, which is the unicellular organism, the genome project of a nematode. It is very important. Here we have completed the genome project of a nematode called Caenorhabditis elegans, which is a non-pathogenic nematode. We have completed the genome project of this nematode. Along with these lower organisms, we have completed the genome projects of animals such as Drosophila, fruit fly. In plants, we have completed the genome projects of rice, maize and Arabidopsis thaliana, 4 o'clock plant. So these are all the non-human genome projects to which we have already completed. The base for this non-human genome projects is the Human Genome Project. By doing this non-human genome project, we came to understand the natural capabilities of these organisms, so that we can use these natural capabilities of these organisms in the fields such as healthcare, agriculture, energy production and environmental remediation. How can we use these non-human genome projects in solving the problems of these fields? The best example is the rDNA technology. In rDNA technology, what we have to do? We have to isolate a particular gene of interest and we have to insert this into a vector, and after that we have to introduce that rDNA molecule into the host to get many copies of DNA. This can be possible whenever you sequence the genomes of that particular organisms using in RDNA technology. If you don't sequence the genome of that particular organism using in RDNA technology, you cannot do RDNA technology. Here in healthcare industry, now we are producing a large amount of insulin by using bacteria as a factories. That is possible due to RDNA technology. Thus, RDNA technology is possible due to the sequencing of genomes of human being and bacterial cells. And in agriculture also, we are producing many genetically modified crops. This production of genetically modified crops through RDNA technology is possible due to sequencing of genomes of those organisms. And the next application is the development of new approaches in the biological research. With the advent of this Human Genome Project, here we have now a new branches in biological research. Those are bioinformatics, which is a new branch of science which uses computational programs in solving the problems in biological research, which is developed parallelly with this Human Genome Project. Likewise, pharmacogenomics is another new branch of science developed due to this Human Genome Project. So the Human Genome Project enabled us to develop new approaches in biological research. And the last application is orchestration of the chemistry of life. If you know the sequence of nucleotides of a group of genes involved in the expression of RNA molecules of a particular tissue, then you can come to understand the coordinated function between this RNA transcript, and you can come to understand the coordinated work between the genes involved of that particular tissue. Likewise, by knowing the sequence of nucleotides in all the genes of an organism, you can come to understand the coordinated work between cells, tissues, organs and organ systems like that. So which means by knowing the sequence of nucleotides in all the genes of an individual, we can orchestrate the chemistry of life. We can understand the biology of life. This is what about the Human Genome Project. In this, we have discussed the introduction, the why Human Genome Project is said to be as a mega project, goals, methodology, salient features and applications. This is all for today's video. In the next video, we will come across the last concept in molecular basis of inheritance, that is DNA fingerprinting. Until that, keep watching all my videos. Please like and share the videos and subscribe my channel Vidya Amrutam Ashnute.